Okay, so today we're going to start in on our first community in terms of our oceanic communities. And our first one is going to be the one that you're probably most familiar with, and that is intertidal communities. If you've ever been tide pooling as an adult or as a kid, or you've just been walking along the beach and seen, some of these organisms, you've been to an intertidal community. And so this is actually a picture of when we would take students down to Puerto Penasco. So some of you that have been to Puerto Penasco might recognize that shoreline. And we would do some tide pooling there. And then these are a couple, this is just an intertidal species called a fiddler crab that we'll talk about when we get to especially our mud flats and estuarine communities. And then this is what a Rockyshore community looks like. So there are different types of intertidal communities based on the substrate. And so we're going to talk about those different types of intertidal communities. This one, as you're going to see, is going to focus mostly on rocky shore communities that have a rocky shoreline. Versus then the next set of lectures will be our soft bottom communities with, your sandy beaches, your estuarine communities like your mud flats, and then we'll eventually get into some really fun stuff like the mangroves. So when we talk about the intertidal zone, you'll also hear it referred to as the littoral zone. More kind of amongst scientific groups, you'll hear them call it littoral zone. So we want you to be familiar with both terms. Intertidal and littoral zone are interchangeable. They're the same thing. And all this means is this is the zone, that narrow kind of zone along the shoreline that lies between the highest high tide mark here and the lowest low tide mark here. Okay, so, and that means that, the very high tide, even if it's only for, a few days out of the year, and the very low tide mark, again, even if it's only for a few days out of the year. So that's your intertidal zone. And so I'm not going to go over what creates the tides again, but I put this up here just as a review for some of you if you needed to kind of review the fact that the tides are created again mostly by the moon, but also by the orientation of where the Earth is in terms of the moon, in orientation to the moon and the sun as well. So remember that we've got spring tides and neap tides, and our spring tides are going to be when the Earth, the moon, and the sun are all in a line, whether it be a new moon here or a full moon, they're all in a line, and this is going to create our greatest tidal fluctuation. This means we're going to have our really high tides and our low tides. And so this is going to be when you want to go tide pooling. And the reason is that's when you're going to get your really low tides. So it's going to allow you to go farther back into the intertidal zone, where you're going to get to see more marine organisms. Now, our neap tides are, again, when the Earth and the moon and the sun are at right angles to each other. So this is going to be your first quarter moon, and this is going to be your third quarter moon. And again, neap tides are not the greatest time to go tide pooling because you won't get to go as far down into the intertidal zone, and you won't get to see quite as many organisms. So if you're going to take your kids tide pooling, you're going to take your family tide pooling, or you yourself, if you're like me, just like to go tide pooling, then you typically want to try to pick spring tides. Again, this is just kind of the best time to go tide pooling if you have that choice. And again, this is just a review of your spring tides again, new moon, full moon, and then your neap tides are going to be first quarter and third quarter moons. And then again, you want to pay attention if you're tide pooling in an area to whether or not you have, the semi-diurnal tides. So you'll have 2 high tides and 2 low tides, and when those times are. If you have mixed semi-diurnal tides, so again, one low tide might be lower than the other, and then or whether or not you just have diurnal tides. So again, meaning just 1 high tide and 1 low tide per day, per 24-hour period. So what we're going to really focus on is kind of some community interactions and the ecosystem. We're going to talk about things at the community and the ecosystem level for our intertidal communities. And so one of the important things is some of the abiotic factors that intertidal organisms have to contend with. So intertidal life is really hard. And not to be dramatic, but it is. It's really hard for the organisms that live in this environment. And part of that is just they have to deal with water fluctuations. They have to deal with being immersed and exposed, right? So that's a big challenge for marine organisms. They have to be able to deal with the tide when the tide's out. And with that comes temperature changes, drastic temperature changes. If you're stuck in a tide pool, that tide pool can heat up pretty rapidly in the summertime, off of the coast of California or down in Mexico. And so you have to be able to deal with these really wide changes in temperature. Wave action. So again, when the tide does come back in, you have these waves crashing down upon you and ripping you off of the rocks. So again, that's another big thing to contend with. Changes in salinity. So again, if you're stuck in a tide pool and it's a really hot summer day, you're going to get a lot of evaporation. And remember, as water evaporates, it's going to leave that salt behind, and you now have a really saline environment that you're trapped in this tide pool. With that, dissolved oxygen becomes an issue. So in that tide pool, as the water heats up, then it can't hold as much oxygen. Also, all the organisms stuck in that tide pool as it heats up are going to start metabolizing faster because their body temperature is warmer. And so they're going to start to use up more oxygen more rapidly. So oxygen, the amount of oxygen dissolved in the water, becomes a limiting factor. And we call this DO. I know we didn't do this lab because we had to kind of trim things a little bit to fit in the 8 weeks. But there was a lab that we usually do when we measure the amount of dissolved oxygen in water. And one of the things that you learn is that the warmer the water is, the less dissolved oxygen it has, the less oxygen it can hold. So literally, because those water molecules are bouncing around, taking up more space, right, as they're bouncing around, there's less room for oxygen to fit. And so warmer water is always going to equate to less dissolved oxygen. And that has to do with the fact that warm water, two factors. Warm water holds less oxygen, and two, the marine organisms in there are metabolizing more rapidly and using the oxygen faster. There's two hits to dissolved oxygen when water temperature goes up. pH. So you're going to have to deal with pH fluctuations. So again, especially as wastes build up in that tide pool, the environment can become more acidic. And so that's going to be a challenge that marine intertidal organisms have to deal with. Nutrients. So the availability of nutrients is going to change depending upon whether or not the tide's out or in. So obviously when the tide's out, you don't have, that water coming in to bring in new nutrients. And then substrate is going to be a really big abiotic factor here. So remember, these abiotic factors are all kind of these non-living factors that intertidal organisms have to contend with. And so this type of substrate is important for classifying what type of intertidal community we have. I kind of mentioned this already. So we're going to focus on our rocky intertidal communities. These are going to be what we call our Rockyshore intercommunities. And so again, our substrate here is going to be rocks, right? Pretty easy. Our soft bottom intertidal communities are going to be in the next set of lectures, and it'll be things like our mud flats, our sandy beaches, and then eventually things like salt marshes and mangroves, and seagrass beds. So those are all going to be our soft bottom intertidal communities. So the big important thing that I want you to get from this slide is that marine organisms in an intertidal zone really have a lot of abiotic factors to contend with. So life is pretty hard in an intertidal zone. Life is definitely not easy for these marine organisms. And there's a video that kind of reviews all this. If you'd like to watch it, it's a PBS video. You can watch it on your own time. I'll also post it under my extra resources for this week. And it's pretty good. It's only about 9 minutes. I'm not going to show it here because, again, my lecture's ready to get too long. But it's pretty good because it kind of reviews all this, but it also shows some video of this as they're talking about all of these abiotic factors that these organisms have to contend with. And so in the video is taken of an intertidal area off the coast of Hawaii. So, of course, nice footage there. So please watch that if you get the chance. It's only, I think, 8 or 9 minutes, so it's really short. Okay, so rocky shore communities are what we're going to focus on. And most of your rocky shore communities, again, we're going to find these on steep coasts that are not going to have the amount of sediment buildup because they're so steep. So, for example, you're going to find this in terms of, if you're looking at North America, you're going to find this where we have our active margin. And our active margin, remember, is going to be off of the coast, the west coast. So we're going to find this mostly on our active margins where we have more recently uplifted or geologically young coasts, or on coasts where erosion, again, removes the sediments and you're just left with kind of the soft rocks behind. So those are the types of places that you're going to find this. So again, like I said, in the US, rocky shore communities are more common on the west coast because we have an active margin. Remember, the east coast is a passive margin. And so you're going to have the steeper rocky shorelines on the west coast where sediment wasn't able to build up because it's such a steep shoreline. And then on the east coast, you'll see this kind of north of Cape Cod. So again, just because of some of the geological features in that area. But most of the East Coast, you're going to have kind of the more sediment, soft bottom type communities because it is a passive margin. So, and then of course, when you have areas where they're just geologically young, you're going to have rocky shorelines. So when you talk about the island of Hawaii, again, Hawaii, especially the Big Island, is very young. You actually have plastic flows currently coming from Mount Kilauea, which is the active volcano there. And you can actually go to, these are pictures I took from Volcano National Park, and see some of these lava flows, which is pretty impressive, especially if you go in the evening. And so these are, brand spanking new shorelines. So much of the coast of especially the Hawaiian Islands, but the Big Island especially, are rocky because it's just geologically young. Again, they're being formed currently by these successive lava flows into the sea. So the coast really hasn't had time yet to accumulate that sediment. And so you'll see rocky shore communities there. And that's why that video, if you go back and you watch this video here, this video is going to be of the rocky shorelines in Hawaii. Okay. So again, and if you look at this picture, this is actually me. And I was actually in Hawaii for a conference there. And I know it's rough life, right? But here I'm standing with someone else that I attended the conference with. And you can see the lava flows behind us, which was pretty amazing. So again, rocky shore communities. When you talk about a rocky shore community, the types of organism that you're going to have here are going to be mostly epifauna. So these organisms, again, are mostly epifauna. And so the reason is that you have rocks, right? And so and just, I don't know that I've gone over this term. I probably haven't with you guys yet because you haven't had this in lab. But epi means on top, like you can think of like epidermis, like your skin. So epi is going to be on top, and fauna is just your animal life. So your epifauna will be your animal life that's on top, okay? So let me see if I can get I have a new pen system here so that I can write. So epifauna, oops, there we go. Epifauna. Epi is going to be on top. So this is going to be your animal life, sorry, and fauna is going to be animal, okay? So this is going to be your animal life that lives on top of the rocky shoreline. So an epi is going to be on top. So the other term that we're going to use is in fauna here. And in fauna, as you can guess, just means that these organisms live in the actual substrate. So it's either on top of the substrate, epifauna, or in the substrate is infauna. And you can probably already guess that your soft bottom communities are going to be more in fauna. Okay. Now there are a few rock boring clams that have the ability to do that, and they are considered in fauna because they're boring into the substrate. But again, not very many. The vast majority of the organisms that you're going to talk about and see in Rockyshore communities are going to be epifauna. So that's a really big, important distinction. You for sure will see that on an exam somewhere. Rockyshore communities, the majority of the organisms are A, epifauna, B, infauna. So you want to know that it's going to be epifauna for your Rockyshore communities. Okay. And so these organisms have to have adaptations to deal with specific abiotic factors for their substrate, for the rocky shoreline substrate. So we're going to talk about all of these different factors, including things like how they attach and how they are able to occupy space and things like that. So we're going to get into all that. One of the things you do have to know for any intertidal community is this special term here, immersion time. So immersion time simply means your time out of the water. Okay, so immersion is the exact opposite of immersion. So you guys probably know the word. Oh, I do so much better with this bamboo, don't I? Oops, well, I said that too soon. Sorry. So I spoke too soon now. There. Let me go back here. As I wasn't paying attention to where I was writing there. So let me go back up here. Okay, let's try this again. Immersion is going to be you're basically immersed in the water, right? That's the easy one. If you can't read my writing, it's IMMERSION. So immersion is your time under the water, okay? And immersion is going to be your time spent out of the water. So it's really important that you understand those two terms, okay? Because we're going to talk about that in terms of when we talk about zonation, how much immersion versus immersion time. So again, be really careful with those two terms. Immersion, okay? And my trick for you to remember this is immersion is your time out of the water or exiting the water, okay? So you can think of exit, exit, immersion. So it's your time outside of the water, okay? And this is going to increase as you go higher in the intertidal zone. So as you're higher up in the intertidal zone, you're going to have to deal with things like simply just drying out. So desiccation is a big one. And desiccation, again, is just kind of a fancy term for, when you are drying out. Because, oops, wow, I thought I was good with this. I'm running off of my pad there. But drying out is one of the big challenges. So again, we're going to use the term desiccation. And there are going to be all kinds of strategies that organisms are going to have to deal with desiccation. So one of the main strategies is you can simply run and hide. Find a wet spot, wait for the tide to come in, okay? So you can find a wet spot in the tide pools. You can find a wet spot in between the byssal threads of mussels. You can find a wet spot in between the cracks of rocks. Wherever you can go to find a wet spot, and then wait for the tide to come in. And then the second one is to clam up. And this means just like it sounds. If you're a bivalve, if you're a mussel, or if you're a clam, you're going to go ahead and close yourself up inside, right? Or even if you're a snail, you're going to take that operculum and close that opening up so that you are completely sealed off so that you can hold that water in until the tide comes back in. Okay, so let's talk about some of these physiological adaptations. I'm going to change off my pen here if I can, maybe. It gives me the option, maybe not. So what are some of the physiological adaptations? And these are adaptations within the organism's internal physiology. How do they use physiological adaptations to deal with these challenges of immersion? And again, being out of the water. So chitons, this is amazing. Chitons, which are these organisms right here, if you see, this is a chitin. They are a mollusk, if you remember, and their shell is broken up into these 8 plates. So you'll see chitons, again, if you're in the tide pools, especially off of the West Coast, you're going to see tons of chitons. And these guys are really well adapted. And the reason that you see tons of them is that they can actually survive up to 75% water loss in their tissues, which is amazing. They can actually, as we'll talk about in a minute as well, they can actually secrete some mucus when they attach themselves to the rock, and that helps to seal off their kind of outer edges and hold in water as well. But pretty amazing that they could survive up to, can you imagine if we could lose 75% of our volume of water and still survive? It'd be amazing, right? So to me, that's amazing. That's why chitons can survive so well in the intertidal zone. Some intertidal seaweeds, certain algae can withstand up to a 90% water loss. 90%. So again, another big physiological adaptation to desiccation here. Limpets actually have an opening that can't be completely shut. So what they do is they will clamp themselves tightly to a rock and seal off that opening by producing a mucus, a thick mucus to make a better seal. And then we'll talk about some behavioral adaptations that they do, making home scars in just a minute, so that when they secrete that mucus, it really seals them off quite well on the rock. So limpets, you'll see these guys, and you cannot pull these off. Same with chitons. Good luck. You see them in the intertidal zone, and the tide is out. You could try, but you're probably going to damage the organism because they're clamped down so tightly with that big muscular foot. They're suctioned down, and then they've secreted that mucus to really seal themselves against that rock. And they are really hard to pull off because of that. So just leave them be if you see them there. Their life is hard enough, right? They're living in the intertidal zone already, so their life is already challenging. Again, algae and rockweeds, remember that these guys have this great physiological adaptation. Many of them, especially rockweeds, can withstand a 90% water loss. So pretty amazing. And we'll talk a little bit about, I have another slide on rockweeds a little further in the lecture here. But many seaweeds and a lot of the sessile algae, meaning the algae that can't move, are limited to living in kind of moist areas that are going to hold a little bit of water all the time. They can't move to one depression, from one depression to the next at low tide. So because of that, most of them are going to exist in these kind of moist depressions. Again, other than rockweed is going to be really successful a little higher up in the intertidal zone because it can withstand so much water loss. Behavioral adaptations. So we're going to talk a lot about periwinkles, especially when we talk about upper intertidal zones. But periwinkles can actually go out and seek microhabitats. I have a picture of a periwinkle, I think, up here. Here's some periwinkles. So these guys are periwinkles. Here's some periwinkles for you here. And so they're going to go and they're going to seek crevices, or they're going to seek, an area where there's a bunch of mussels, a big group of mussels that have a lot of byssal threads that are going to help to hold water in that area. So when the tide goes out, they're going to seek those microhabitats, and that's going to make them one of the more successful organisms in the upper intertidal zone, the fact that they have the ability to do that. Also, they can clamp, they can close off their operculum, take their operculum and close themselves up and clam up and help to hold in water. So that's another adaptation, one of the physiological adaptations they have. But between the physiological and the behavioral adaptations that periwinkles have, it makes them very successful in the upper intertidal zones. Limpets, this is really cool. Limpets actually do this interesting behavior called, they make these home scars. And what they do is they take their shell and they kind of twist it. They use their big muscular foot and they twist back and forth against the rock until they scrape it with their shell. Sometimes they'll use their radula, but as you can see, most of them will use their shell. And they'll scrape it back and forth until they create this little home scar. And then that gives them a better seal on the rock when they actually secrete the mucus around there. So you'll see this on some of the rocky shorelines. You'll see these home scars in the rocks, which is kind of interesting. And so these guys are the limpets. We call them keyhole limpets, and you'll see them scattered throughout the intertidal zone, especially in the upper intertidals. And limpets typically will remain in one spot at low tide. And then when the tide comes back in, they'll graze on algae. They're a grazer. So again, you can see some of them will also migrate just like the periwinkles to seek microhabitats and, amongst the byssal threads of mussels and things like that. But if they can't, then they'll go ahead and use this behavior of making their own home scars and suctioning themselves down. Some of the dominant organisms, again, of your rocky shores are going to be periwinkles. We talked about those. So you can see what periwinkles, I gave you some pictures so that you can see what periwinkles look like. And again, the success of the periwinkles, I already talked about this because I kind of jumped ahead of myself, but the fact that they can, again, go seek microhabitats, but then they can also clamp themselves to the rocks and seal in moisture. And then they can also simply just close up this operculum to seal off the opening of their shell. So I believe I brought these out in lab, but if I didn't get a chance to, these are called sand pennies, and these are actually the operculum. And so what a snail will do is this is attached to its muscular foot, at the end of its muscular foot. So it'll pull itself into its shell. And once it pulls itself into its shell, it'll use this operculum to seal off the opening here. And it does that, again, to help hold in the water and prevent itself from desiccating. So, and then again, they have that behavioral adaptation where they can congregate in moist, shady areas. So those are kind of the three ways in which periwinkles can prevent themselves from drying out. And this makes them really extra successful in upper intertidal zones. So you're going to see periwinkles are found kind of throughout the world on these rocky shorelines, different species, but they're found in the upper intertidal zones throughout the world because they're so successful at these three things to prevent desiccation. Mussels anchor themselves to the rocks with these really strong byssal threads. And we'll talk about this is important for, again, preventing them from being ripped off with wave action. But they'll also group together like this. And when they group together like this, it actually helps to hold water a little bit better. So that provides, when the tide's out, a more moist environment. And then they oftentimes, higher up in the intertidal zone, mussels will actually have thicker shells to prevent water loss. And then they're going to form these nice dense clumps, like we just talked about, to help retain moisture. And again, they're going to use that clam up strategy. So close those two valves and clams themselves up. Salinity. So again, salinity can become a problem in the intertidal zones, and especially in a tide pool, for two reasons. So again, you can end up having a bunch of evaporation from that tide pool, leaving the salt behind, and it becomes really saline in that tide pool. And then you can have the opposite problem, where if you get rain, that can be really detrimental to intertidal organisms. Again, remember, these are marine organisms that have evolved, again, to a saline environment. So then you can have the opposite extreme where you get too much fresh water input from the rain. And so again, it's really tough for these organisms because they're adapted for a marine environment, not a freshwater environment. So too much rain can literally kill intertidal organisms in a tide pool. So this is where a clam up strategy is helpful to prevent that fresh water from getting into your tissues and causing those osmotic imbalances, right? And then again, like we talked about, these intertidal organisms have to deal with both extremes. So then there's going to be these hot, dry days in the tide pool where water is evaporating off, leaving the salt behind, and then the tide pool is going to be really saline. So most of your tide pool organisms have to be adapted to deal with this wide range in salinity. Okay, really challenging for all these marine intertidal organisms. Many of them have restrictions in terms of the amount of time that they can feed. So again, if you are talking about your rocky shore communities, little sediment can accumulate there. So deposit feeders are really rare here because there's no sediment and no deposits for you to feed on, right? So you don't see deposit feeders here. Most of your sessile animals here are filter feeders, but you can only filter feed when the tide's in, right? And so you're limited, especially how high up you could be in the intertidal zone by your feeding time, because again, when you're immersed, when you're outside of the water immersed, you can't filter feed. So filter feeders, grazers, and many of the predators aren't going to feed, obviously, when the tide's out. That's not a problem for your animals in the lower intertidal zones, but obviously higher up in the intertidal zones, a lot of these organisms are going to grow slower because they simply have less feeding time. They have less time immersed under the water to feed. So this is going to limit some of the organisms that you have from moving higher up on the shoreline, okay? And this may seem, you may say, well, okay, well, why don't all the organisms live lower and down, right, in the lower intertidal zone? So we're going to talk about why that is in just a minute here. So intertidal life is not always easy when the tide comes in either. Sorry, I'm taking a quick drink of water here. And so there's going to be all kinds of adaptations that organisms are going to have to deal with wave shock. And this is as the tide comes back in, you have to deal with wave action, right? And so if you are a sessile organism, you have to have ways to deal with wave shock. If you're a mobile organism, you can use some behavioral, right, techniques. So you can go tuck underneath a rock or in a crevice. And so you'll see some of your intertidal fish would do that. You'll notice that a lot of your intertidal fish are small and elongated. And part of this is just physiologically, it allows them to be and to move into little crevices, especially like when you're talking about blennies. They'll move into little crevices, or gobies into holes in the sediment. So that's one of the reasons why most of your intertidal fish will have these kind of elongated bodies. Also, gobies and clingfish will have these kind of modified suction cups that are formed by their pelvic fins. So when you look at, you can't see their pelvic fins here, these are their pectoral fins, but their pelvic fins underneath will be modified to either kind of prop themselves up on the bottom, or they'll also be modified as a big suction cup, which I'm going to show you a clingfish in just a minute. And you'll see this big modified pelvic fin. And it acts as a suction cup to suction themselves down to the rocks so that they can battle the wave action. Limpets and chitons are just simply going to use that big muscular foot, and they're going to suction themselves down. You also probably notice that limpets and chitons have this nice low profile, right? So they're not sticking up and having to fight against the wave action with this large profile. Also, your intertidal fish, oftentimes they're going to lack swim bladders. And we haven't talked about swim bladders yet, but these are these big air-filled bladders that you're going to see inside fish when we dissect them. And they use those big air-filled bladders for buoyancy, to control their buoyancy as they're swimming. But many of your intertidal fish just lack that swim bladder altogether, so that they just sink and stay in the bottom. They don't need to be buoyant. They're staying in this intertidal zone. So that's another adaptation you're going to see in some of your kind of mobile animals. So many of them are just going to move, like I talked about, and find shelter when the wave action is too strong. So a little bit of an easier way of dealing with it than if you're a sessile organism. So here you can see this is, if you look here, this is this this modified, and this is called a clingfish. Oops. This is that modified pelvic fin, and it acts literally as a suction cup. So that's what you're going to see in your clingfish. So that's a pretty awesome adaptation in terms of adjusted for intertidal life. Adaptations to cope with wave shock. So sessile organisms have to deal with wave shock as well, except for their problem is they can't move, right? They don't have that behavioral modification. So they have to just deal with the wave shock. And they have to have some physiological adaptations usually. One of them is by simply just having a very strong attachment. So you'll see this in barnacles. Barnacles secrete this glue-like substance that is really strong. And so that allows them to really attach very strongly to the rocks to prevent the waves from ripping it off as those waves come back in. And I'll talk about an article with this in just a minute. But yeah, so they secrete this glue-like substance. And it's pretty cool because we'll talk about some biomimicry and things that we're trying to create based off of this glue-like substance that barnacles secrete. Seaweeds are going to have something called a holdfast. So we're going to get into this more when we get into the kelp and algae section in lab. So we'll do this and you'll get to see these holdfasts, but it's a lot of times people think seaweeds are a plant and that they have roots, and they don't, because their roots wouldn't have anything to dig into, because you're going to find seaweeds on rocky shore communities. So they actually have what are called holdfasts that are going to attach them to the rocks. So we'll see some of these, and I'll show you some of these in just a few minutes here. And then mussels are going to produce these byssal threads that you saw in lab. And these are these really strong proteinaceous fibers that will attach themselves to the rocks. So strong attachment. And again, you want to make sure you know these ways to cope with wave shock. So one of them is by producing a really strong attachment if you're a sessile organism. The second one is to simply have a thickened shell. So a lot of your mussels in areas that have a strong wave action are going to have thickened shells. Same with your barnacles. Having a low profile so that you are, again, closer to the rock and you're not meeting as much of the resistance from the wave action is another big adaptation. And so we'll talk about this in sea anemones in just a minute. We're actually going to talk about each of these. Or again, if you don't have a low profile, you can just be flexible and kind of be able to move with the wave action. So you're not fighting against that wave action. So we'll talk about that with sea palms. So first, again, we're going to kind of talk about each of these really briefly. We'll talk about, each one of these and give you some examples. So strong attachments, again, barnacles and seaweeds are great examples of this. Mussels are great examples of this. Seaweeds are going to use these holdfasts to encrust themselves onto rocks. So they're going to, here's the holdfasts that I was talking about here. And you'll see, again, they look like, because a lot of people don't know that seaweeds are actually algae. They're not plants at all. So they look like they have roots, but those are not roots. They're actually simply a structure called a holdfast that does just what it sounds. It holds that algae fast to the rock. So the main function of that holdfast is to attach that algae to the rock. And it's going to secrete a glue-like substance as well from each of those holdfasts. Mussels, again, are going to hold onto rocks with their byssal threads. So they'll have, a lot of byssal threads that maximize their surface area. Same with the holdfast here, really maximizing their surface area to attach to as many places on the rock as they can. And then barnacles are going to secure themselves to the rock with this glue that's so strong. Several companies are really trying to duplicate it. And I have posted this article for you. Please read it. It's really interesting. I'm going to pull it up here real briefly for you. It's a fairly new article, came out just in August 2021. And they are actually currently doing research on barnacles. And they're trying to create this paste based off of this is what we call biomimicry, where we're trying to mimic the substance that barnacles use to glue themselves. And they're trying to come up with a compound that's similar to this to stop heavy bleeding for medical purposes. So right now they're doing it in rat and pig models, and they're looking at some of the like long-term impacts of it to make sure that it doesn't damage tissue over time. But this is eventually something that you could actually see in the medical community. So a really interesting biomedical engineering use for barnacles. And so in terms of when we talk sometimes about conservation of of some of these marine organisms, people might say, well, why do we care about, preserving marine intertidal organisms? Why preserve these? Why are they important? And the reason is that many of these organisms contain this chemical biodiversity within them. They contain chemicals that, we as humans don't produce, or we can't even find out there. And that chemical diversity can be important in terms of potential uses for us in terms of medicine and other products. And so when we lose some of this biodiversity because of habitat destruction or climate change or whatnot, along with it, we lose some of this chemical diversity that we have within our organisms. And that loss of chemical diversity can truly impact us in terms of the fact that we might not have the capability of using that down the line if we needed it. So again, this is one of those great examples of how marine organisms really can directly benefit you. So let's see. When we talk about, and we'll get back, sorry, my aside there. So that's strong attachments. Again, how organisms strongly attach to a rocky shoreline. The next adaptation for dealing with wave action is simply have a low profile. So again, anemones, especially the giant green sea anemone, and you guys have probably seen these if you've been out off the coast of California and been tide pooling there. You'll see these, the giant green sea anemones. And they're really pretty. Many of them have the zooxanthellae, so they have a symbiotic relationship with a photosynthetic organism. So they're really brilliantly green. And these guys are going to live in kind of your low intertidal zones, so a little farther down in the intertidal zone off the Pacific coast of North America. But because these are rocky shorelines and they're fairly steep, many times they do have to deal with some wave action. If they're growing in sheltered locations, you'll see they're going to be much taller. But if they're in areas with heavy wave action and they're not very sheltered, they're going to typically be much shorter. And that's because this is going to reduce the body area that's exposed to that wave action. And it's going to allow more water to kind of slide over the top, helping to reduce drag. So that's a very common way. And it's pretty cool because these are the same species. So this is just a physiological adaptation to the specific microhabitat that they're living in. Again, if you're in an area that's sheltered, then you'll see a taller form of the same species. And in an area that's less sheltered with more wave action, you'll see a lower profile. Again, but the same species, just a physiological adaptation. The other, again, way to deal with wave action is just to be flexible. So sea palms are a great example of this. And so this is one of your sea palms. It's a type of brown algae. And it withstands the waves by just being flexible. When the wave action comes in, the currents are high, its fronds are simply going to fold up, and it's going to streamline the plant and reduce that water resistance against the wave action as it comes in, as the waves hit. So again, really a very simple strategy in terms of instead of meeting the resistance of that wave, it's simply going to just streamline and reduce the water resistance across it. So real quick, how I give that away. What is the greatest limiting resource? Harder to do. I don't get to ask you these questions because I'm recording these lectures. But if you were here, I would say, what do you think the greatest limiting resource for rocky intertidal organisms are? And most people are going to say food, right? But actually, that's not really the case for your rocky intertidal organisms because food is pretty plentiful. You have quite a few plants and algae that are able to grow. It's shallow enough, they can get plenty of sunlight. So you actually have a lot of primary producers in your rocky shore communities. Also, the tides are constantly bringing in plankton. So again, sometimes drifting seaweed, even sometimes detritus, right? So there's plenty, and remember, just as a reminder, detritus is just particles of dead organic matter, okay? That detritus feeder is gonna feed on, especially things like crabs. We'll talk about, there's some crabs that are big detritus feeders, and they're gonna feed primarily on detritus. So again, the really primary producers, you've got lots of them. So food is not really a limiting factor here. Oh, and just as a reminder, deposit feeders are really rare here. Why is that? Because there's no sediment here. Our substrate's rock, so we don't have any deposits to feed on. So our big factor, limiting factor here is just going to be space. Believe it or not, space to attach to is your biggest limiting resource. So key thing here, you need to know this for the exam. I will tell you this will be somewhere on the exam, that because this is going to be different, obviously, for your soft bottom communities. So your limiting resource, your largest, greatest limiting resource for rocky shore communities is going to be space, okay? Make sure you know that one. And so sessile organisms, again, organisms that can't move, need a place to attach to. Real quick, I just wanted to show you this before we get into this, because we were talking about, there's plenty of primary producers here. So you're going to see again, you've got your base of your food web here, your primary producers, seaweeds, diatoms, seagrasses. Diatoms are just going to be single-celled algae. And again, you're also going to have, plankton that are brought in here. Here's plankton that's going to be brought in with the tides for your filter feeders. And so then you're going to have, as some of this breaks down, it's going to form detritus in the seaweeds and the seagrasses, and then you're going to have scavengers that are going to feed off of that. You'll have grazers that are going to feed directly off of things like your seaweeds and your primary producers. You'll have filter feeders that are going to feed on plankton, and then you'll have all kinds of, again, carnivores that are going to feed on these organisms here. So that's kind of your basic rocky intertidal community structure. And so your carnivores, you're going to have sea star snails, fishes, crabs, flatworms. And we'll talk about this when you talk about your upper intertidal zone. Your other challenge for your upper intertidal zone is that you're going to have land predators. You're going to have things like birds and rats, raccoons, humans, right? Because we come collect things like mussels. So you'll have actually land predators higher up. But this is just kind of your real basic food web. So let's get back to this battle for space. Again, nearly all space of an intertidal zone is going to be occupied. And so you can see here's a little clearing, and eventually this space will be occupied by either these gooseneck barnacles or these acorn barnacles or these mussels. So all space, almost nearly all space in your intertidal zone, especially as you move kind of lower into that mid-intertidal, will be occupied eventually. So competition for space strategies, okay? One of the easiest ones is just simply to be the first to get to the open spot. And to do that, remember these organisms are going to be using larval dispersal. So they're all going to be, especially these sessile organisms, are going to have a larval form. So that means that you have to be really effective at dispersing your larva. So, or spores if you're, we'll talk about in things like some of your sea palms. Sea palms are going to release spores. Then again, the second competition is to colonize an already occupied spot. So push out whatever is there, and then colonize that spot. So barnacles can actually do this as they grow. They can grow underneath the neighboring species and undercut them, loosening them from the rock. And then as a wave comes along, a really strong wave, it'll pull off that organism. So that's one way that you can deal with that, is colonizing already occupied spots. Sometimes, again, other organisms are able, we'll talk about how they can rebuff spots. When you would talk about sea palms, we'll talk about one of their special strategies for colonizing an already occupied spot. And it actually involves sacrificing kind of their first brood, their first group of spores. It's an interesting strategy. And then the other strategy, the third strategy, is just to simply grow over your competitors and make them really vulnerable to waves by smothering them or blocking sunlight. So again, sea palms kind of use this strategy, and we'll talk about that they use both of these strategies by sacrificing their first group of offspring. So this is how they do it, and a really interesting strategy. Sea palm adults will actually release their spores nearby, surrounding them. And those sporelings will grow on their competitors, especially things like mussels and barnacles. And those sporelings, as they grow up, will provide a larger profile, right, than what the mussels, the byssal threads of the mussels or the barnacles are adapted for. They're not adapted for this larger profile. So as the first rounds of some really heavy waves come in, they're going to rip off those sporelings along with the competitor, whether it be a mussel or a barnacle. And that's going to clear space for the next generation of young sea palms. So they kind of sacrifice their first generation for the second generation here. So they really kind of use both the second and third strategy there. Okay. So because, again, there's such this competition for space, we're going to talk about what causes zonation in our Rockyshore communities, and why don't all organisms inhabit the lower intertidal zones where they're immersed and they don't have to deal with, desiccation, and they don't have to deal with the extreme temperature changes and salinity changes. So we're going to talk about that. And so we end up, if we look at Rockyshore communities, you actually are going to see these bands on our Rockyshore communities. And a lot of times you'll see a band of barnacles, especially the steeper it is, the more apparent that zonation will be. And so this is actually a picture I took from California, off the coast of California here. And you can actually see, if you look really closely, you can see, some of these smaller acorn barnacles kind of in between some of the rocks here. And then you'll see there's a band here of gooseneck barnacles. So if you look, here, there's this band of gooseneck barnacles across here. I'm not going to draw in here because it'll just take too much time. And then here, there's your gooseneck barnacles. And then here, there's a band of mussels. Really distinct zones here. And that would just happen to be, this was actually a really steep area. I was looking down from up here. And so the zonation was really, really super apparent. So that's what you're going to see in these Rockyshore communities. And obviously, if you have a more gradual rocky shoreline, this zonation will be more spread out. So barnacles were actually studied to answer this question of why we have zonation. And so we ended up using what's called the little gray little gray barnacles here. And we found that these little gray barnacles, and you'll see them right here, they're found mostly in your kind of uppermost parts of the mid-intertidal zone. So they're going to be higher up in the intertidal zone. And we'll talk about what this mid-intertidal zone is in a little bit, but they're going to be found higher up in that intertidal zone. While larger species of these barnacles, like your rock barnacles here, are going to be found lower in the intertidal zone. So down where they're going to be more immersed, okay? They're going to spend more time underwater. So upper intertidal zone is going to be, again, towards the shoreline, and lower intertidal zone is going to be, again, towards the waterline or immersed under the water. And so what they did was they ended up doing a series of experiments. And what they found is if they allowed each of these species to just disperse their larvae, to see where they disperse their larvae without any other factors impacting them, they found that there was quite a bit of crossover in terms of the areas that they could disperse their larvae from. So when they looked at larval dispersal here for the little gray barnacles, even though you typically only find the little gray barnacles in this small band here, they found that they could disperse their larvae much farther down, lower in the intertidal zone. And they found this the same with the rock barnacles as well. They could disperse their larvae both higher up and lower in the intertidal zone. So larval dispersal wasn't the big factor here in terms of limiting where these organisms were surviving. So then they started to look at some of the factors that would limit life higher up in the intertidal zone. And the biggest one, we're going to talk about this, are going to be more of your abiotic factors. Your non-living factors are really going to be what limits life in the upper sections of your intertidal zone, each intertidal zone. And so here it's really going to be immersion time, your time out of the water, desiccation, drying out. And so what they found is each of these organisms, the little gray barnacles, couldn't survive, even though their larvae could disperse up in this area. They simply couldn't stand the desiccation. They would just dry out and die. Same here. Now, if you look with the rock barnacles, which are larger, they couldn't withstand this area here because being larger, they would simply dry out quicker. And so they couldn't withstand this area that the little gray barnacles could. So that allowed the little gray barnacles to actually inhabit higher areas of the intertidal zone than was physically possible for these rock barnacles. Now, again, the big question is, why don't all organisms inhabit these lower areas of the intertidal zone, right? Where they don't have to deal with desiccation. And this is where biotic factors come into play. Things like competition and predation. And those are going to be the two big factors that are going to limit the lower limits of an organism's intertidal zone. So here, when we looked at the little gray barnacles, we found that competition was heavy between the little gray barnacles and the rock barnacles. And the rock barnacles were simply just a better competitor. So again, that limited the little gray barnacles to this area because the rock barnacles would outcompete them. Now, the rock barnacles, being the better competitor, okay, were then limited farther down by predation. So as they moved farther down, they became, again, subject to predation by dog whelks, which are actually, it's a predatory snail. I think I have a picture of one here, yeah, which they look, right, so innocuous, but they actually have a radula that can drill into the barnacle and feed on the barnacle. So predation by dog whelks was a factor down here, as well as competition with mussels that live down here in this lower intertidal zone. So those became limiting factors for the lower limits of these organisms. So this really helped us establish why we get this vertical zonation. It helped us establish that in the upper limit of every zone, there was typically physical or abiotic factors, right? Abiotic, remember, means non-living without bio, living, so non-living factors. So things like desiccation, temperature changes, salinity changes, things like that. The lower limit of each organism's distribution was determined more by biological factors, things especially like predation and competition. So this, again, slide, you can mark this, is really important because this is why we have vertical zonation, okay? So you need to know these two factors or these two limitations. What limits the upper limit of an organism in its zone? Again, abiotic factors like desiccation, again, like temperature and salinity changes, things like that. And then the lower limit is really going to be determined, again, by biological factors, predation and competition. Okay, so make sure that you know that. This one kind of shows, this was from an example of the impact of mussels that mussels have in terms of, again, impacting zonation. And In this case, we're going to talk a little bit about your keystone predator. I know we've already talked about this, so I'll just go through this briefly, and that's sea stars. So again, mussels' upper limit is typically set by desiccation and feeding time, these physical factors. And then their lower limit, so here's your band of mussels right here, right? And so this was as far as they could grow up just because of desiccation and feeding time. Again, when the tide's out, they can only move so far up in the intertidal zone because then when the tide's up, they can't feed. So they were able to live in this band here. But what limited them from moving farther down was predation by sea stars. And sea stars really need to be able to feed when the tide's in. So this really limited, again, predation by sea stars into these lower intertidal areas. So what ended up happening is when you removed all the sea stars from an area, the mussels would simply just outcompete and competitively exclude all of these other organisms, creating this monoculture of mussels. And that really, again, limited the biodiversity in that area. So in this case, our sea stars, remember, are our keystone predator. And so we'll talk about this in a minute. This really goes along with what we call our moderate disturbance hypothesis. So this is going to be a second example of another keystone predator. Spiny lobsters actually also prey on mussels in some areas. And again, spiny lobsters are going to be lower in the intertidal zone because that's where they're going to feed. And so what they did was they created these cages. And some of the cages were open and would allow the spiny lobsters to come in and out. And the other cages were closed and would prevent spiny lobsters and other potential predators on your mussels. And what they found is, again, just like here when they removed the sea stars, when they did this, we ended up with a monoculture of mussels, okay, mostly mussels here. Few barnacles are still existing, but mostly mussels here. And so again, this is where we have our keystone predators. Our sea stars and our spiny lobsters are going to prey on mussels, and this is going to open up space for other things like gooseneck barnacles and seaweeds. And without those predators there, then it completely decreases the biodiversity in that environment, completely changes those communities to where they are almost a monoculture of just one species. And in this case, it's mussels. So sea stars also eat dog whelks, and dog whelks feed on barnacles. So this gives barnacles a chance to grow big before a whelk actually eats them. So this really adds to the fact that sea stars are these keystone predators in here. So the big key takeaway from this is that keystone predators ultimately are going to increase our biodiversity in this area. And so this plays into what we call our moderate disturbance hypothesis. This hypothesis that moderate disturbance is best. Areas where you don't have competition, don't have predation, are going to be, because you've maybe removed your keystone predator from those areas, are going to have a lower diversity because it's going to allow what we call competitive exclusion. So let me put this here. That's going to allow the better competitors to outcompete the other competitors. So this is going to allow, oops, if I can do this without pushing my buttons here, competitive exclusion. Exclusion here, okay, where the better competitor is going to completely exclude other species. So our moderate disturbance is best where we have some predators, we have some disturbance that is going to keep those top competitors from taking over. Constant disturbance is going to lead to low biodiversity as well, because this isn't going to allow your community organisms to make a foothold, right? Take a foothold. So if you had like constant disturbance between, possibly some new predators that were really, detrimental to bunch of different species in that area, not allowing them to inhabit that new open space. Or if you had something like, back-to-back hurricanes, okay, that's going to lead to, too much disturbance and a lower biodiversity. So moderate disturbance is best, okay? These are disturbances, again, like predation that can give other species a chance to move in. And disturbances can increase the number of species in a habitat if you have moderate disturbance, which increases the overall biodiversity by preventing competitive exclusion, just like we talked about. And the highest biodiversity is going to occur when there's enough disturbance to prevent competitive exclusion, but not too much disturbance that the community is not able to develop. Okay. So one of the last kind of interesting examples of this is this is actually an example of just some biological interactions that determine which types of seaweed actually are going to predominate in certain New England tide pools. And unless something comes along and interferes with this, you'll actually see both types of tide pools are kind of self-sustaining in different areas. So you'll actually find both tide pools in different areas. And the reason being is that in this tide pool here, I'm just going to show you kind of the two examples. What ends up happening is you have this algae called Enteromorpha, and it grows really long and kind of filamentous like this. And it actually will help to hide some of the predators in here, like the crabs, these green crabs, from the gulls, which feed on them. And so what ends up happening is you have quite a bit of this Enteromorpha. You end up having quite a bit of these green crabs, and they eat quite a few of these snails. And the Enteromorpha ends up actually overgrowing what's called the Irish moss. And so it ends up forming what we call this Enteromorpha pool. And it's kind of its own self-sustaining pool. Now, what's interesting is you can move, just a little bit farther down, and you might see a pool that's more of an Irish moss pool. And the reason that you have an Irish moss pool is the Irish moss will mainly grow when the Enteromorpha is gone. The Enteromorpha is the better competitor and will outcompete the Irish moss. But if you have a pool where you don't originally have a lot of Enteromorpha, then the seagulls can actually eat the crabs in the area because they can see them. They don't have the Enteromorpha to hide the crabs. And then because the crabs are being removed by the gulls, there's fewer of the crabs to eat the snails. The snails can then feed on the Enteromorpha and overgraze the Enteromorpha, and that leaves more space for this Irish moss. So again, just a really interesting story in terms of how you end up with these Enteromorpha pools and these Irish moss pools in this kind of same environment in New England. And so again, just very kind of simple food web where the Enteromorpha feeds, or the periwinkle snails feed on the Enteromorpha, the green crab feeds on the periwinkle snails, and the gull eats the crab. And so just again, by just having this slightly different habitats, if the Enteromorpha inhabits this area early on, it creates a very different habitat than here in the Irish moss pool. And the interesting story, the story becomes even more interesting when you talk about the fact that the periwinkle snails won't inhabit new tide pool areas. So really interesting. You think that the periwinkles would end up moving from one tide pool to the other, but they don't actually end up doing that. So you think they just move from one Enteromorpha pool to the other, but they don't. The large periwinkles tend to prefer to stay in one tide pool, and they don't move from tide pool to tide pool. So interesting. And the young, again, periwinkles are eaten by the green crabs. So in this tide pool, it's beneficial for them to stay in there because there, again, aren't as many green crabs because the gulls are eating the green crabs because the Enteromorpha isn't hiding them. So again, just your book kind of goes over this story, and it's just an interesting, really interesting story in terms of how these two different, very different tide pools can exist within the same type of habitat. Okay, so let's go. Again, when we talk about vertical zonation, there are these three main zones. And I'm sorry, I'm going to try to go through this and make this not so long, but this lecture always ends up being longer than I think it's going to be. And so the three main zones that we're going to have are going to be the upper intertidal zone, the mid intertidal zone, and the lower intertidal zone. And we'll use these terms throughout the world for describing our rocky shore communities. The upper intertidal zone is going to be where our immersion time is greater than our immersion time. And so in your middle intertidal zone, sorry, I had an alarm going off here. My middle intertidal zone is going to be where your immersion time is about equal to your immersion time on average as a whole, right? Over a span of a year. Now, when you have something like neap tides, where you have, a really big tidal fluctuation, so you're gonna have times where the upper mid intertidal zone will have more immersion time than immersion time, and your lower mid intertidal zone will have more immersion time than immersion time when you have these big tidal fluctuations, okay? But on average, if you take that whole area, on average, the immersion time about equals out the immersion time, total time over a year. Then the lower intertidal zone, of course, is going to be where your immersion time with an I is greater than your immersion time. So your time immersed underwater is greater than your time outside the water. Again, as a whole, you're talking about over a year. So these are, again, very general descriptions, and boundaries between each zone isn't always absolute. The more steep your rocky shoreline is, the more definitive those boundaries are going to be. And then the more gradual your rocky shoreline is, you'll see those boundaries will not be quite as distinct. So your upper intertidal zone is what we typically call your splash zone. This lies above the high tide mark, and your organisms are going to be wet by wave splash and spray for most of the time. They're not going to be totally, immersed very often. You don't really have filter feeders here because, again, this is your splash zone. They're simply just not submerged long enough to be able to feed. And then your dominant primary producers are going to be things like lichens. I'm going to show you a lichen in just a minute. It's a fungus with a mutualistic algae that lives inside of it. And that algae, again, it can do photosynthesis and produces nutrients for that fungus. And in return, the fungus is actually going to provide a stationary place for that algae to attach to the rock. Then we have encrusting algae and cyanobacteria. I'll show you what these look like. These are your dominant primary producers in the upper intertidal zone. And then your most dominant animal is going to be your periwinkle snails. So in that upper intertidal zone, you oftentimes are going to hear your upper intertidal zone called the periwinkle zone. And that's why. That's one of your indicator animals. Periwinkles are going to graze on algae. They're going to breathe air like land snails, and they can tolerate really extreme temperature changes. They can also live without water for months. So that makes them really well adapted for life in the upper intertidal zone. And that's why you're going to see periwinkles in this zone kind of all throughout the world. Limpets as well. You're going to find them. They're really hardy grazers, and they're going to feed again on things like some of your algae that are in this area. And then you're going to have few marine predators here, okay? So maybe you'll have a few, shore crabs, a few predatory snails that might, like the unicorn snail, that might make its way up into these upper intertidal zones, but not very many. Most of your predators here are going to be land predators. So you're going to have things like birds, coyotes, rats, raccoons sometimes. Marine shorebirds are a big one here. And then even humans, right? Because we do things like collect clams and mussels and things like that. Obviously not in the rocky intertidal zone. We're not collecting very many clams because they're in fauna, but Again, humans are going to be potential predators here as well. So I went ahead and put this kind of picture together of some of your upper intertidal indicator species. So this right here is a lichen. And again, lichens, you're liking the lichen, right? My kids hate that joke. They hate that joke. So lichens, again, are this fungus that will attach to hard substrate like rocks, and it has a mutualistic algae that lives inside of it. So there's your lichens. Here's some of these slimy cyanobacteria, and here you can see this big cyanobacterial green mat here. That will form on the upper intertidal zones. You'll also have some types of encrusting algae that will just encrust the rocks here. And then here's your periwinkles, which are a big indicator species, and then your limpets as well. Here's a limpet. So those are all things, if you see a lot of these species, that's a good indicator that you're in the upper intertidal zone. Okay, we're going to focus mainly on the Pacific in terms of your zonation and your indicator species that will help indicate what zone you're in. But this is the Atlantic. So if you do go to the Atlantic side, you can see some of these species as well and use this as your kind of map for your intertidal zones to tell what zone you're in. So, but again, we're going to kind of focus on the Pacific just for time purposes, and also just for the mere fact that you guys are closest to the Pacific and most likely to visit the Pacific. So again, we've just talked about mostly the upper intertidal zone, where you have periwinkles, limpets, lichens, encrusting algae, cyanobacteria. Then we're going to get into our mid-intertidal zone, and we're going to break this mid-intertidal zone up into the upper mid-intertidal zone and the lower mid-intertidal zone. And then we'll talk about our lower intertidal zone. So in our mid-intertidal zone, again, this is going to be an area that's submerged and uncovered by the tides daily, and sometimes even twice daily if you have the semi-diurnal tides. Zonation within this zone is going to be due, again, to variation with submersion immersion times. So your upper mid-intertidal zone is going to be the area that your total immersion time in a year will be greater than your immersion time in this upper boundary. And even though, again, you're still mid-intertidal, it's going to be submerged and uncovered by the tides each day, but your overall time out of the water in this area will be greater than your time in the water. And so this, you're going to see some smaller barnacles. You're going to see things like acorn barnacles, gray barnacles, and your rock barnacles. So here's some examples of these smaller barnacles. This would be your acorn barnacles. Some of the rock barnacles are going to look very much like this, but just bigger. Okay. And then your lower boundary, or your lower mid-intertidal zone, where again your immersion time over a year, okay, is going to be greater than your immersion time you're outside, okay? And so even though it's submerged, at least and uncovered at least once daily, if not twice daily, your total time in the water, immersion time, is going to be greater than your immersion total time over a year, okay? So in this what we call lower mid-intertidal zone, you're going to find things like mussels, seaweeds, rockweed. So rockweed is a type of seaweed that you'll see right here, and I put this on here so you could get a good look at what it looks like. And rockweed does really well in the lower mid-intertidal zone, and it has these little air bladders called pneumatocysts that help float the fronds a little closer to the light. So here you can see some of these little pneumatocysts or air bladders. We'll talk about don't worry about the term pneumatocyst just yet, because we'll get to that when we get to algae. So in a little bit here. But that helps to float that frond closer to the light, because again, it's an algae, it needs to do photosynthesis. And then also you're going to see different barnacles here called gooseneck barnacles. So they're going to be a larger barnacle, and they have what looks kind of like a gooseneck here. So that's why they call them gooseneck barnacles. And these are your gooseneck barnacles. So you can see they look quite a bit different than your acorn barnacles. So the upper mid intertidal zone will have acorn barnacles. Your lower mid intertidal zone will have more of your mussels and your gooseneck barnacles. And you guys probably remember what your mussels look like. Let me see if I can find some mussels. I'm trying to remember where I had a picture of mussels last. You guys know what your mussels look like though from lab, because we did mussels in lab. So you'll find those in your lower mid intertidal zone. So again, here, primarily upper mid intertidal will be your smaller barnacles, like your acorn, your gray, and your rock barnacles. And then the lower is going to be mainly your mussels, your gooseneck barnacles, and your seaweeds and your rockweeds. And then we'll get into the lower intertidal here, which is where you're going to see seaweeds and all kinds of predators. So lower intertidal zone is really the fun zone. This is where you're going to see the greatest biodiversity, and this is where you want to get to if you're going to be tide pooling, because you'll see the greatest biodiversity in this zone. And the reason is that, again, your immerse time is much greater here. So your immerse time is most of the time. Most of these organisms are spending most of their time underwater. So this is easier for things like predators, like sea stars and predatory snails to feed. Mussels and barnacles will be more rare here, and that's due to predation by things like sea stars and predatory snails, and also competition as well. So you'll see that these lower intertidal zones are going to be dominated by many different seaweeds. So you'll have red, green, and brown algae or seaweeds here. And that will really provide quite a bit of not only habitat in terms of the seaweeds providing a place for organisms to hide and take cover, but it will also provide food for organisms as well. So there are many different organisms that live within the seaweeds, hiding from predators. So you're going to see things like sea urchins and sea anemones, polychaete worms, snails, sea slugs, and many, many more invertebrates. And this is why, again, if you go tide pooling, you want to tide pool in a neap tide so you can get down, because that's the greatest tidal fluctuation. That's when you're going to get your low tides, and you're actually going to get to the lower intertidal zone, where you're going to get to see all of these amazing, species, the biodiversity that you really want to see tide pooling. So again, you're going to see things like here, these big green sea anemones. You'll see lots of different types of algae here. You'll see sea stars. You'll see sponges. You'll see, again, echinoderms. So you might see like your Pycnopodia here, or in Mexico, you'll see what's called your Gulf sun stars that look like this. It basically looks like a sea star with like 20 different arms. And then you'll see lots of sea stars here. You'll see sea urchins and nudibranchs and sea slugs. Again, this is where you're really going to see a lot of your biodiversity. And you'll also get to see some of your rocky intertidal fish. So again, you'll see things like gobies and clingfish that have that modified pelvic fin to attach to rocks, and pricklebacks and gunnels. Again, a lot of these rocky intertidal fish, you don't have to know all of these different fish. We're going to talk primarily about like gobies and blennies when we get to when we talk about marine fishes later on. But what you do want to know is that these guys, again, are going to have these kind of more elongated bodies to allow them to hide in the crevices as the tide moves out, or when the tide moves in to hide from wave action. And then many of them are going to lack swim bladders, so they can, again, just sit on the bottom like this. And then they'll oftentimes have modified pelvic fins to be able to prop themselves up, attach to the bottom, or even suction themselves down. So pretty interesting. So again, most live in this lower intertidal zone when you're talking about the rocky intertidal fish, because they do need to be in water. They need to be submerged underwater to be able to do gas exchange with their gills. So again, those are some of the most common rocky intertidal fish that you'll see. And so again, up next, sorry, this went a little longer than I wanted this lecture to go, but we'll be talking about soft bottom communities, and I'll get into the next lecture will be sandy bottom communities and mud flats on our estuaries. And then our last lecture will probably be mangroves and maybe even some of our salt marsh stuff. Okay, so that's it. I will see you all next time.